Hello students, this is Dr. Faizan Mirza. Here I'm discussing with you a question that came regarding ELISA, a technique that we don't really study at A level, but this is again an assessment objective two question in which they give an extended information, some detail that you have not really studied, but you have the core knowledge to address this and do it very well. So let's navigate this question along. Question number 6. This deals with an enzyme. You very well can recall what an enzyme is, how these enzymes work. So they are saying there's an enzyme HRP, horseradish peroxidase. It is extracted from this thing plant and is used in industry. The reaction catalyzed by HRP, which is the enzyme, is on the substrate H2O2 and is used to oxidize an organic substrate. The figure shows organic substrate plus hydrogen peroxide. HRP produces oxidized substrate. This is the product, and water is produced. Inhibitors can affect the Vmax. We know that. In this reaction, in this graph, they're saying complete the figure 6.2 by drawing a curve to show the presence of a non-competitive inhibitor will affect the rate of reaction. So we know if this is the Vmax, then the Vmax with the non-competitive inhibitor should be way less. We'll find out the half Emax, and with the half Emax, we can find out the Km value. The Km value comes to be precisely 0.06. We mention it together with the unit. You know how to read the information I have given the working here. This is something very basic, which you can do at your own as well. Part B, scientists can design synthetic DNA nucleotide sequences to produce a synthetic HRB gene, which means the gene that can produce this enzyme. These sequences will include start codon and a stop codon. So the translation of messenger RNA can occur. Explain what is meant by start and the stop codon. So start codon is the first triplet of bases, A, U, G, on the mRNA that attach to the ribosome during transition and is called the start codon. It codes for the amino acid methionine, which is the first amino acid. The last triplet of bases could be UAA, UGA, UAG, either one of them. We'll mention all three with the slash to say that one of these will be there. On the amino is called the stop codon. It does not code for amino acid and stops the elongation of the amino acid chain. Here, this ELISA technique is shown. This HRB, which is the enzyme, I have annotated this to just make sure that it becomes easier to follow. This enzyme is used in an immunological test known as sandwich ELISA. ELISA stands for enzyme-linked immunosorbent assay. We perform this technique to identify various chemicals in our body, any hormone and such as anything which is of a specific shape. So how sandwich ELISA is used? One use of this test is to diagnose a disease. Figure 6.3 outlines the main steps in sandwich ELISA in which a toxin is released by the pathogen. We know the toxin must be an antigen. It is attached to it is detected in a sample of body fluid taken from a person who is ill. The test involves two types of monoclonal antibody. We know what antibody is. An antibody is having a something like this structure. This region is constant region and it attaches to the neutrophils, whereas this region at the top attaches to the specific antigen because it's having a shape complementary, which is the variable region to the antigen and can attach there. The monoclonal antibodies are there. They can bind to toxin, a capture antibody and a detection antibody. So two different kind of antibodies will be used here, a capture antibody and a detection antibody. Step 1, prepare a test well by mobilizing the capture molecule monoclonal antibody to the support surface. So this is a support surface, a capture antibody is attached. This capture antibody constant region is attached here. And this region here must be the region which will be complementary to something that can attach to. This is the test well. Remove a sample which will be having antigen from the fluid of the person and add it to the sample well. So you add it here. The toxin gets attached here. So why the toxin gets attached? The toxin gets attached because the toxin is complementary to the antibody here. So this antibody is having a binding site complementary to the antigen. It attaches to the antigen. Whereas this antibody fails to attach. Why? Because this antibody is not either complementary to it, or even if it's complementary, so the toxin probably was in low quantity, less concentration. So it got attached only to two of them, not the third. Wash out the test well, remove any substances not bound to capture antibody. So the capture antibody and antigen have formed a complex. We can see it here. Add the detection antibody to the test. The detection antibody is the one which is in a reverse, you can say the shape in which this is the antigen binding site and the constant region Here, the constant region is attached to this star-shaped thing called HRP, which is the enzyme, same horse reddish peroxidase. So this detection antibody is added. Wash out the test well to remove any detection antibody. Add the substance H2O2 catalyzer that can catalyze HRB and add acid to stop the reaction further. Complete the figured the situation after step 5. So when this antibody is added, this antibody being detection antibody will come and attach to the toxin. When it comes and attached to the toxin, so this becomes a sandwich. This is what the sandwich ELISA basically means. The toxin gets sandwiched between two antibodies. One antibody held at the surface, one antibody having a detection marker, and the toxin or the antigen held in between. Why did we not attach the antibody here? This detection antibody is not attached here because the detection antibody variable region is not complementary to the variable region of the antibody. It is complementary to the antigen. So both the antibodies have a variable region complementary to the antigen. So that's why they both attach here. With reference to figure 6.3, describe one difference between the capture antibody and the detection antibody. Capture antibody is immobilized, while detection antibody is mobile. Detection antibody is having HRP attached, while the capture is not. The reaction is shown to you that TMB is produced. This compound is the organic substrate. TMB is a colorless compound. They are saying that it reacts with H2O2 in the presence of the enzyme turns blue-green and water. So if this blue-green product is forming, it means more of H2O2 is being actually reacting there, and this enzyme is working more. So if a lot of blue-green color is forming, it means that the colorimeter will show you increased absorption, which means decreased transmission because it's a colored product. A student suggested that a low concentration of this toxin, which means the antigen, can be diagnosed by a negative result, can be diagnosed as a negative result, which means it's absent, instead of a positive result, which means it's present. So if there is a very low quantity of this antigen, there may be a case of misdiagnosis where the antigen gets missed and it's not diagnosed. Using a colorimeter after step 6 in Sandwich ELISA test would provide a quantitative result and help avoid this error. How so? Quantitative result means you are using colorimeter to get the numerical reading. We'll have the less colored product. Less colored product means there will be low absorbance, high transmission, and you can relate to it that even if there is a minor change in the absorption or transmission, it means the product is there. With reference to figure 6.5, explain why using a colorimeter would provide a quantitative measurement for detection of a low concentration of toxin and help to confirm a positive result. So the colorimeter can measure the absorbance and transmission. A lighter colored solution gives a low absorbance, which means more light gets transmitted, which shows less quantity of the substance being analyzed. In this case, this thing, TMP, the blue-green color. HRP bound to detection antibody binds with the toxin, which is already attached to the capture antibody. So when H2O2 reacts with HRP in step 6, it produces the colored product, which is blue-green TMB. If less toxin is attached, less detection antibody will be attached to the capture antibody, and less HRP is available to react with the hydrogen peroxide. So less quantity of blue-green TMB will be produced. This small color change may still not be detected by the eye or by conventional method. This may be subjective, but a colorimeter will give you a precise numerical reading even if there is a very low concentration of this product, the colored product. The transmission or the absorbance can detect these minor changes in concentration. So yes, this was a 10-mark question. Yes, this was a difficult question on surface, but again, it's testing your core knowledge of antibody and colorimeter and how the enzymes work. So 3 things integrated together. I hope this video was helpful for you. That's all from my side. Thank you so much.